Hi, my name is Bement, and I'm from the University of Wisconsin at Madison. I'd like to tell you a little bit about a study of ours, the results of which were published in the August issue of Developmental Cell. The work was done by Brian Burkel, a man of many talents and many ensembles. Brian had help from Alain Benink, Emily Vaughn, our collaborator George von Dassow, and me. The system employed for this study is frog oocyte wound healing, and the idea is pretty simple. The oocytes are wounded, actin filaments and myosin II accumulate around the wound, forming a contractile ring. The ring then closes over time. What directs formation of the contractile array? The small GTPases, Rho and CDC42. These proteins are activated in concentric rings or zones around the wounds and then close inward in concert with a contractile array. Contractile rings are not restricted to wound healing. The most famous example of a contractile array is provided by cytokinesis. The so-called cytokinetic apparatus is made of actin filaments and myosin II and controlled by Rho. Our starting question was simple. Why do contractile rings close inward? It's long been thought that contractile rings close by virtue of myosin-powered movement. The myosin pulls on the actin filaments, shortening the ring. Surprisingly, when we inhibited myosin activity, the contractile rings still closed. Let's talk about the experiment. The first thing you need to know is that we have three means of inhibiting myosin. We can shut off Rho, we can shut off ROCK, or we can shut off the myosin itself. The second thing you need to know is that we can quantify the effects of myosin inhibition using cortical flow as a readout. Cortical flow is powered by myosin II and results when the myosin II pulls on actin filaments. The actin and material associated with it moves toward the myosin in the site of contraction. The movement of cortical flow resembles that of breakfast cereal sprinkled into a stream of water. We can use brightest point projections to generate a single image of the process of cortical flow. In such images, cortical flow is revealed as streaks of dots converging on the site of contraction and myosin activity. Let's look at the results. In control samples, a ring of actin forms around the wound and closes inward. The closure is accompanied by robust cortical flow, indicative of myosin II activity. In contrast, when Rho is inhibited, while the ring still forms, there is no cortical flow, showing that the myosin II is inhibited. Nonetheless, the ring closes inward. And it doesn't matter how we do the experiment. If we inhibit ROCK or inhibit myosin II directly, we nonetheless get rings forming and closing in the absence of cortical flow, and thus myosin II-powered contraction. Further, it's not just actin, but the ring of myosin and the zones of the active GTPases also close inward in the absence of contraction. This is very strange. The contractile ring and the GTPase zones close inward in spite of the absence of inward movement of their constituents. How can we explain this? One possibility is by what we call the signal treadmill hypothesis. The analogy here is to cytoskeletal polymers such as actin filaments or microtubules. These polymers can, under the right conditions, polymerize at one end and depolymerize at the other simultaneously. Likewise, we envision that the Rho GTPases may be preferentially activated at the leading edges of their zones and preferentially lost at the trailing edges of their zones, thus producing the illusion of movement. The recruitment of active Rho and CDC42 to the leading edges of their zones is consistent with the treadmill model. Ideally, however, one would like to observe the inactivation of the GTPases as well. This was accomplished using photoactivation. Photoactivatable probes for active Rho and active CDC42 were generated and introduced into cells. Following wounding, cells are illuminated with a 405 laser to activate the probes. The spatial pattern of probe disappearance reveals where Rho and CDC42 are being inactivated. Analysis of the pattern of probe loss relative to the position of the contractile ring showed that Rho is indeed preferentially inactivated at the trailing edge of its zone. Analysis of the pattern of probe loss also showed that the preferential trailing edge inactivation becomes more pronounced over time. Thus, a contractile array is backed by a GTPase signal treadmill. It will be of interest to see if treadmills are used in other contexts, such as cytokinesis.